Okay, let's just start with the DNA. Currently, we know DNA is a genetic information. Genetic information means the information we can transfer to our offsprings, which can define our shape and personality and characteristics, all traits. So those information should be stored in some material and then transferred to the offsprings. Those informations are stored in DNA. To us, it's quite obvious maybe, but some time ago, it was not really obvious which material is really genetic information. The main candidate of this genetic material was either DNA or protein, and there was some historical experiment which can determine which one is really genetic information. Before I show you the result, would you stop the video and think to yourself, if it was your problem, how would you solve that problem? How would you figure out which is your real genetic information? Can you design that kind of experiment? Okay, let me show you the answer. In 1952, two scientists, Hershey and Chase, they designed a really smart experiment. They chose this bacteriophage, the virus, as model system, and this virus has very unique structure. It has outside material as a protein coat, and then it has the genetic material of DNA in the head here. So when they infect the bacteria, the virus binds to the surface of the bacteria and then inject DNA material inside the bacteria here, leaving the protein coat outside. Now, utilizing this bacteria, they labeled the protein with the radioactive material. So now, only the protein is radioactive and then DNA is normal DNA. Then, when the virus bound to the bacterial surface and DNA is injected inside this bacteria, and then they can form new virus particles inside. Now here, which one is radioactively labeled? This protein coat. And if you shake this bacteria to remove this bacterial coat, then they found that there is no radioactive material inside here. Now this time, they changed the experimental format. This time, now they labeled this DNA with radioactive P32, and they repeated the same experiment. Then, the virus binds to the bacterial surface. Now this DNA is injected this bacteria, and then they removed this bacterial coat, the protein coat, and then they synthesized the new virus particles. Now the radioactive material is incorporated in this the bacteria. So by this experiment, it clearly shows that the DNA, not the protein, is a genetic information material which can transfer the traits of a virus into the next offsprings. Quite smart experiment, isn't it? After confirming DNA is the genetic information, the next year, 1953, Watson and Crick, they proposed the structure of DNAs. So this is the famous double helical structure of DNAs. DNA is a double helical structure like this. And if you open up, now two strand DNA connected to each other. Assuming that we open them up, now it's a two strand and finding its partners along this line. And then they are condensed to make this helical structure. And DNA is a highly negatively charged molecule because of those phosphate group. That's why to stabilize those other charges, we need some counter partner with strong positive charges. Now the partner here, this is histone. Histone is positively charged protein, and then DNA molecule is wrap up one complex of histone molecules, and then this one unit we call nucleosome. And then different nucleosome can combine together to make is even bigger the clusters. And this is supercoils and find another partner and then make up to the even bigger coils. And eventually, in middle state of the cell status, they are dispersed form of the DNA, so-called chromatin structure. But when the cell divide into two, then they combined together and make this highly condensed form of DNA, so-called chromosome. So now in the cell division stages, we can see even those chromosomes under the light microscope. And then two copies of chromosome is aligned in the middle of the cells. And then eventually they move to each end of those cells. Now this is the centrosome for each new cell. And then the cells divide into two daughter cells. DNA has four building blocks, and those are A, T, C, G. And then depending on those connection the directions of this deoxyribose the position, this is 5' and this is 3'. So from the 5' end to 3' end we read. And these two strands, by matching these two the base pairs, it's anti-parallel directions. One is coming from here and the other is coming the other directions. And this deoxyribose is connected by a phosphate bond. That's why DNA is highly negatively charged the molecules. And this base pairs has its own unique partners. A is matching with the T with the two hydrogen bonding. And then depending on those directions, this side is the big groove, the major groove. And then inner side here, this is a connecting side to the ribose. And this is a minor groove. In the same manners, and G is matching with the C with a triple the hydrogen bonding. That's why usually CG bond is considered to be stronger than AT bond here. Same way, this side is a major groove and this side is minor groove. And the distance between these base pairs are parallel to each other, and this distance is approximately 3.4 angstrom. And the 1 ton is composed of about 10 base pairs. The total length should be about 3.4 nanometer. And the diameter of this double helical structure is a little bigger than 2 nanometer size. Now if you look at here, the DNA structure, this side is major groove and the other side, opposite side, is a minor groove, right? Now, if you look at here, A and T are the connection. Now, can you tell which side is major groove? The key point is this base connection to the sugar. This is ribose connection here. So, connecting side here, this side was minor groove, right? And this side should be major groove. Same way, here, this pyrimidine is oriented here, and this purine is oriented here. So, this is minor groove, and this is major groove. Now this major groove and minor groove is connected to opposite direction here. DNA is this shape of double helical structures. If you think about the DNA synthesis, the nature utilizes highly energetic triphosphate structures. So ATP is known to be very common biological energy source or the biological battery. Now here, similar materials, DNA case, we don't have the hydroxyl group here. This is DATP, deoxy ATP, and this is a building block for the A site. Similar way, CTP is utilizing this, the deoxy CTP, deoxy GTP, and deoxy TTP. These triphosphates are the energetic the building blocks for the DNA synthesis. When DNA is growing, now here this is a 5' and this is a 3'. 3' OH is a nucleophile, and then new ATP or GTP coming here, and then this hydroxyl attack this alpha phosphate, leaving pyrophosphate here, making new bond of phosphate here. So in this case, the energetic intermediate is this triphosphate structure. So we can say the direction of DNA synthesis is from 5' to 3' directions. And this is the DNA replication scheme. DNA is a double helical structure, That's why, to make it a copy, first of all, that double helical structure should be divided into single strand first. So the enzyme participating in this dissociation is called as a helicase. Once it's opened up, each strand can act as a template for the synthesis of new strand of DNAs. Now, if it's the case of here, now in this strain, this one is a 3' end to the 5' end, then synthesis can happen from 5' to 3'. Now, this is very smooth the growth. That's why this strand can be synthesized continuously to this direction. How about the other one? Now here, the open strand is 5' to 3'. Now it has some problem because the direction of synthesis should be from right side to left side. Then the opening is the start here. Then it's not full strand is open. Now each fragment should be synthesized separately, and then later it should be connected here. So the process was suggested by Japanese scientist, and that's why this small fragment here is named following his name, Okazaki fragment. Let's look at the dynamic movie for the DNA synthesis. This double helical strand is the original DNA. And then by help of a helicase, it's divided into two strands. One strand here, DNA polymerase here, it's synthesizing from 5' to 3' direction. It can go continuously. But the other direction is synthesis should happen from 5' to 3'. It cannot make a continuous growth. That's why it should make a one loop. This fragment is called Okazaki fragment. Now synthesis is moving through here and then finishing up here. It makes one fragment and then starting another fragment here coming, and then connection happens here. Again, this is a continuous synthesis, and then this is a fragment synthesis, and then connection happens in this site here, connected and removed. From different angle, again, this is a continuous synthesis, and this is Okazaki fragment, and then connection happen here and then leave, and then start another fragment synthesis. One more angle, again this is a continuous, and this is fragment synthesis, and move around. Quite interesting, isn't it?